Chromosome structure, function, and nomenclature. Chromosomes are long thread-like structure. We all know about chromosome. We are students of biological science and medical science. Okay, so chromosomes are known to us. It's a long thread-like structure composed of DNA and protein called histone. Okay, each chromosome consists of a single continuous DNA molecule which contains many genes. There are thousands of genes on each chromosome, and roughly we have 20 to 23,000 genes in our genome. The main function of chromosome is to carry genetic information necessary for development, functioning, and reproduction. So that means whatever information is present on chromosome, it is as it is inherited to the next cell, that is the daughter stem, after the completion of cell cycle, and to the next generation. Okay, and that is the overall idea, to preserve the genetic information that we have. Now we have to keenly understand the structure of chromosome so that we can understand the procedure of karyotyping. Chromosome has three important parts: the telomere, that is the end of chromosome; the centromere; and the arms, that are the P and Q arm. Telomeres are like the end cap of chromosome. The main function of telomere is to protect the chromosome from fusing with another chromosome, and also protect it from deterioration. Okay, that means it is very important part of a chromosome to like align its structure precisely. Otherwise, if telomeres are not there on the end of chromosome, okay, chromosomes can fuse together and it can like mess up the genetic information. Okay, mostly telomeres contain repetitive nucleotide sequences and it does not contain any functional genes. There are a couple of reasons behind that. The one is the end replication problem, we can say. Whenever the replication start for a chromosome, or we can say for all the chromosomes, when it reaches to the end of chromosome, that is the telomere site, uh, the replication process will slow down, or sometimes it miss the sense of replication, and that means whatever sequences are present on telomeres, sometimes they cannot, we can say, correctly replicated, and that means if important genes are present on telomeric side or on the cap of the chromosome, those genes cannot effectively replicated and cannot be transmitted to the daughter cells. And this is the reasons the genes are present on the arm, that is on P and Q arms, and not on the telomere. However, the repetitive nature of telomere is also important for us. Okay. I guess if you heard about the role of telomere in aging, telomeric sequences have substantially important role in the aging process. Okay. As the telomere gets shorter and shorter, because it like less prone to replication, and it gets shorter and shorter of we age, and that means as we age, the telomere get shorter, vice versa is true. So it is very important to protect our telomere in a very good strength to avoid aging process. Okay, now moving ahead, centromere is the another important part of a chromosome, we can say. It's a central part, but not exactly the central part that divides the chromosome in equal sizes. It's a central part that joins the sister chromatrix of chromosome, we can say. The arms joins the arms. It's a very constricted area. The DNA, the repetitive sequences that are present on centromeric region are very tightly packed. So no enzyme can involved in the process of transcription, and that means the nucleotide sequences, we can say the DNA that is present on the centromeric part cannot participate in the transcription process effectively. The P and Q arms contains mostly the genes, and that means they are known as the gene rich regions. Okay, but it also contains non-coding DNA as well. Okay, the short arm of a chromosome is known as the PM, and it is derived from the word French word that is peptide. And following that, the Q is coming in the alphabet order. So the another arm is known as the Q arm. Okay. So this is the short introduction of chromosome, we can say. Now understand the hierarchy of how the chromosomes are formed. Okay, we have a nucleotide sequences. Everybody aware about the nucleotide sequences? that is the we can say A, T, G, and C nucleotides that along with the sugar and phosphate make the DNA, construct the DNA. That DNA interact with the histone protein and make the nucleosome like structure, then the bead on a string like structure, and then the chromatid and then chromosomes. Okay. So chromosome is a complex network of protein and DNA. Now why DNA has to pack on chromosome? That is important to discuss. Okay. See, uh, not every DNA that is present in our genome is coding. Okay, that means there are 23,000 genes. Hardly 3% of our genome can make any protein. So those DNA part that should be loosely available so that other enzymes can bind there and performs the gene expression activity. Okay, transcription or translation. Okay. So, DNA is packed in a way that are accessible during this some stage of cell cycle for transcription and translation. Okay. Other than that, other DNA are very tightly packed on a chromosome that cannot allow any enzyme to participate in any DNA metabolic activity. Okay. And ultimately, it forms the thread-like structure that we know as the chromosome. Chromosomes are situated in a cell, particularly in the cell nucleus. Now, the structure that we know as a like thread-like structure is only appearing during the metaphase stage of the cell cycle. Okay, most of the time, what happens in during interphase or other stage of cell division, it is present in a loosely we can say DNA structure that doesn't have any particular structure. Okay. So if we want to study the chromosomes, if we want to know the structure and number of chromosome that is present in a cell, we have to arrest the cell. We have to we can say lock the cell, particularly on the metaphase stage, and that is the main objective to perform the karyotyping. In the karyotyping procedure, what we do is we arrest the cell on the metaphase cell cycle stage so that we can study the chromosomes. Okay.